Hi everybody, so I've got this paper I'm working on, Effect of Different Light Intensity on Physiology, Antioxidant Capacity, and Photosynthetic Characteristics on Wheat Seedlings Under High Carbon Dioxide Concentration in a Closed Artificial Ecosystem. Mouthful aside, we've got 4 scientists out there in Beijing, China, working for what I presume is the Chinese space industry. They're working in Lunar Palace 1. It's basically a glorified apartment, but it's simulated space where people aren't allowed to go back in and out, and they hold some solid world records of people artificially going into space with it. Now we're looking at the wheat. What choice of light exposure you use, that's pretty important. You're going to use certain amounts of light. It's going to affect how the plant grows, how well it does or how well it doesn't do, how well it uses water, because you're in a space environment, with an expectation. Lower light and less efficient means, and you're not just going to be doing open air. You have an environment with a high carbon dioxide concentration. And the breed used here was spring wheat. So with the high carbon dioxide concentration, that's the big goal here in this experiment. And their study aims to reveal potential mechanisms for the physiological and developmental responses to different light intensities on wheat seedlings in a high carbon dioxide concentration environment. And there's supposed to be a hypothesis to every experiment, and that's kind of something I'm starting to realize is that's not quite how papers work so much anymore. Officially stated, there's not a hypothesis, but the hypothesis you could pull pretty solidly from this is that the amount of light exposure affects plant growth and the plant development and characteristics regarding the plant. Now it's a little bit broad, and I guess it's a little easier instead of saying, oh, it's going to affect this and this and this and this and this. They just threw a goal at it. Now, the experimental groups involved were 6 different light intensities. You're putting more light out there, more light onto your plants. All of these exposures had a 24-hour period, and they all had the same light. There wasn't something with a little bit different wavelength. The one variable they were using in all of this, in the exposures of 200, 400, 600, 800, 1000, and 1200 micromoles of photons per square meter per second. Now to give you some reference, the sun gives out between 900 to 2000 during the brighter parts of the day. So these are overall on the pretty low end of where sunlight's going to be at. But some of that's being made up for with the high concentration of carbon dioxide. Now the experiment, and this I feel like was a little bit of a flaw, because if you're sitting there trying to test specifically how well is this wheat going to feed these astronauts under these light conditions, why not go all the way till you have food product? That would be like, go all the way and check it out. But they stopped early for the 20 days. It's good preliminary experiment for future studies, but really it does need to take it to the next level, because there are some issues with the experiment that I believe are going to have long-term impacts. So they were going for 20 days, right? Height measured every 2 days. There were a bunch of different chemical tests and sensor tests, and these are the things they checked for. You got the membrane stability index. That's basically the durability of your cell. If this is low, cell's got a higher chance of rupturing. You've got a lot more issues with your lipids in your membrane, and a lot of those are stemming from free radicals, oxidation, stress, peroxides, that kind of stuff. RWC is your relative water content. That's more like how much water these cells are storing. If they're storing more water, that's less watering that needs to be done. That means more water is saved. Peroxidase activity is pretty important. Increased peroxidase means more protection against that oxidative stress I was talking about earlier, those free radicals, peroxides. Those all are occurring as byproducts of photosynthesis, electrons jumping around the cell doing their various things. So more peroxidase is a good thing. On the other hand, MDA is not a good thing. Malondialdehyde, if that is high in concentration, that suggests there is a lot of oxidative stress. Oxidative stress means tissues are being damaged because oxygen's moving around, because there's free radicals bouncing. You're taking lipids, you're tearing them up, you're ripping out hydrogens and slapping in oxygens and doing all sorts of things. You're going to have a little bit of rearrangement. It's not good for the cell. It's not good for the lipid. Proline's another enzyme. It does a variety of functions, not perfectly proven, but among one of them, it's like fighting off pathogens and some other things. The big thing for this experiment, it tends to take peroxides and break them down into safer compounds. Another thing they were checking for, pretty easy one compared to everything else, chlorophyll analysis. How much chlorophyll is in there? And we're talking about two types here, A and B. More chlorophyll is a good thing because you're going to be doing more efficient photosynthesis. And another point of the photosynthesis efficiency was a little more the testing of heat side. Is a lot of photosynthesis energy being wasted because it's turning into heat? Chlorophyll fluorescence is, again, another thing regarding the efficiency. It's something highly fluorescent. That means there's a lot of stuff bouncing off of it. That means there's a lot of energy not being. So the results, the height of the wheat was almost identical in every plant, regardless of the exposure. That surprised me. I thought more light was going to make growth faster. I thought it was going to make it bigger. Not a difference. Really surprising, especially with those really low levels of light. Now the membrane stability and the relative water content were very similar in every test group. There was like one or two that were really smaller than the others, but they were all really similar. So basically they're all similar durability cell-wise and similar water storage. Now the 600 micromole group was the highest in most of the observed results. Not all the results were good. MDA, for example, not good. You have high MDA, that means there's a lot of oxidative stress, a lot of tissue damage is happening that'll accumulate, destroy organelles over time, even result in plant cancer. Now the MDA results followed a curve. I'm talking our middle values we were testing earlier, those 600, 800, those values were higher. 400 was a little bit high. 200, 1000, 1200, those were pretty low. Peroxidase was a similar situation with our middle values having high values, or middle experimental groups having high values, outer ones not so much. Photosynthesis efficiency, this followed a really solid upward trend. The more light exposure, the more photosynthesis efficiency. Now the 600 experimental group contained the most chlorophyll A and B, and the 1200 group contained the second most chlorophyll A and B. And the fluorescence results were similar with group 200 and 800 scoring the worst. So almost across the board it was basically the same, but 200 and 800 did a little worse. So there's really nothing popping out on top on that one. Now to hop over to the graphs for us real quick. Watching the straw height, although there's a little bit difference here at the beginning, kind of you start doing the recordings and things are going a little faster, they all came down to pretty much the same height. Relative water content, got error bars that put these things well within each other's range. And the membrane stability index, all pretty similar. Peroxidase activity, 400 pulled far away and ahead. That's good because it's going to be protected pretty well from a lot of that oxidative stress. There's sort of a bit of a curve going on there, but it's pretty weak. MDA, we've got 600. So again, this one scored the highest on a lot of things. It's doing fairly well for a variety of things. But it is not doing well in the MDA department. It's going to have a lot of stress, a lot of damage, a lot of mutations adding up. For our photosynthesis, pretty strong trend going on here. Intrinsic water efficiency, very important if you're in a spacecraft because water is a very limited resource. 600, less watering, less water usage. Chlorophyll content, again, 600 and 1200, it's got the highest. Some of these other photosynthetic things were pretty even across the board. Some of this is the fluorescence. We got 600, it's doing pretty well. Let's talk about the conclusion and some of the stuff that was pretty interesting here. The study determined the 600 micromole light exposure is most suitable for high carbon dioxide concentration wheat. I mean, if you go back and look at the results, it wasn't like it blew away, except for in the water conservation. It didn't really blow anything else away. And it also had the high MDA, which it had the worst on that. You know, that would be definitely not a good thing. They must not have valued that too bad, though. But MDA, like I said, it's a sign of high oxidative stress and damages tissues. You know, if you're having chloroplasts being damaged over time, and you're having lots of mutations build up, you also get an inconsistent product. MDA is also mildly toxic and mutagenic. It's got some not so good stuff in there. It can damage things. MDA, we'll take a quick look. Malonodialdehyde. It's highly reactive. You don't want anything highly reactive inside of a cell. You want it to be mild. You want everything to be easy. So this is another thing where I'm asking about the full fruition. Why not wait for the whole time? Why not wait till you can see flour, dough, whatever you're going for? I'm not sure the best way to bake bread on a spaceship, but what I do know is if you're having a lot of mutations, a lot of damage to the tissues, they could build up. They can get your wheat. If MDA is building up in there, that's also a little bit toxic. That's not something you want to be feeding your astronauts if you can avoid it. So getting something low in that would be a good call. So when the scientists jumped pretty quickly to the conclusion that the 600 was the best choice, I was kind of like, it didn't score that well in the other stuff. It's got good chlorophyll. That is a big deal. So it's going to be producing sugar. It's going to be producing those carbs. It's going to be producing food. Some of that stuff needs to be evaluated. It needs to be seen all the way through. Can't judge a book by the first chapter. But that was their decision. All right, everybody, have a good one.